translated into proteins. We're going to focus on transcription and translation. As we go, we're going to start with a gene, transcribe it into an RNA molecule, and then translate it into a protein. That's what gene expression is all about. One gene codes for the building of a protein. Here's an overview of both transcription and translation in a bacterial cell and in a eukaryotic cell. We're going to focus primarily on the eukaryotic cell because that's just where we're headed. And I have some words here some questions here that we're actually answering, but I want to look at the picture while we're answering them. So you can be writing things down on this slide, if you will. So first of all, if we're looking at a eukaryotic cell, we see the cell. This maybe is a plant cell. I'm not sure. It looks kind of like it has a cell wall, right? Yeah, it has a nucleus, and in the nucleus is the DNA. Right? All of the DNA is in there. A piece of the DNA is called a gene, right? So maybe this is one gene. And in transcription, a piece of DNA, a gene, is transcribed into a single-stranded RNA molecule. Okay. Well, what are genes? Genes are segments of DNA, strands of DNA that have a particular nucleotide sequence and that code for the building of a product, usually a protein product. They code for the synthesis of a protein product. In transcription, then, DNA is copied, this piece of DNA, a gene, is copied into RNA. Well, what is RNA? Ribonucleic acid. It's also a nucleic acid, just like DNA. It's a little different, right? Remember some of the differences between RNA and DNA? RNA is made up of ribonucleic acid, so it's got a different sugar than DNA. It's got ribose instead of deoxyribose. RNA has 4 nucleotides, 4 different nitrogenous bases, but they're not exactly the same as DNA. Remember, DNA's bases are A, T, C, and G. That's DNA, right? What are the 4 bases for RNA? A, is there a U? No. Is there a T? No, there's a U. Uracil instead of thymine. C and G. These are the bases for RNA. So there are one difference in the bases, and RNA is single-stranded. So we get a single chain of nucleic or nucleotides, okay, making a single-stranded nucleic acid. But we say that RNA and DNA use the same language because they're both nucleic acids. And there's a faithful copying of DNA to RNA, a faithful copying. So what essentially we do is we make an exact copy of the DNA. Why do we think we need to make an RNA? If I'm trying to use a gene to make a protein, why have RNA at all? It's like the middleman. Why is it necessary? This is a good question. We're not even sure of the answer. Some people might say, well, you need to have RNA because the DNA stays in the nucleus up here, and you need to make RNA so that it can leave the nucleus and then go on to translation. But in bacterial cells, if we look carefully at our bacterial cells, we don't have a nucleus. The DNA doesn't stay in a nucleus. It's there it's out in the cytoplasm. And so we're not exactly sure why RNA is necessary. We know we have it. We know it's used. There might be an evolutionary history here that we need to think about. And it's a pretty interesting question that a lot of evolutionary biologists study, actually. Why is there RNA? Okay. All right, so now we've made RNA. And actually, here's our RNA molecule. And it looks like in eukaryotes, it goes through some sort of processing phase before the RNA leaves the nucleus and heads out to the cytoplasm. In bacterial cells, there's no RNA processing that happens. Okay, the RNA is made and then it's used in translation. Now, in translation, the mRNA molecule, that's this guy right here, right, moves into the cytoplasm and is translated into a new language. The new language is amino acids. The RNA is used to make a sequence of amino acids. And at this point, we usually call the RNA molecule the messenger RNA molecule, the mRNA molecule, okay? And we make a sequence of amino acids. Those little purple dots in our picture are all individual amino acids. So in translation, there's machinery like ribosomes and tRNA molecules and other enzymes and energy that's used to take the nucleic acid code of RNA and convert it into an amino acid code of proteins. That's why we say that we're translating, because we're changing the language here from nucleic acids to amino acids. We're going to look at transcription and translation in eukaryotes. But just to comment, a note here, and we've already mentioned that in bacterial cells, there's no RNA processing. In bacterial cells, there's no nucleus, so transcription and translation can happen in the same place and even at the same time. Okay. All right, we're going to take a look, though, at eukaryotes. Let's focus on them. I think we've answered all of these questions, this overview process. So we're going to start thinking about transcription and what happens in transcription. And here's a picture that we're going to look at, and I'm going to draw a couple pieces of this as well. This is figure 17.8. And what we're looking at here is a piece of DNA. Let's just call this, it's called the transcription unit. Think of this as, being a gene, right? I'm having trouble with writing my word gene there. Notice that the DNA is double-stranded. Okay. The strands run in opposite directions, 5' to 3' and 3' to 5'. And we're trying to copy or trying to make a copy of this gene, right? And so we're thinking about how this happens. In the end, you notice that you have a copy of the gene. That's the red molecule. Our textbook uses color coding here. DNA is blue, RNA is red. And here's what has to happen. We divide transcription into an initiation phase, elongation phase, and a termination phase to end it. All right, and obviously we're going to be using something called Rna Polymerase, because here you see this kind of weird triangle-shaped structure, and it's called Rna Polymerase. Rna Polymerase is an enzyme, and it polymerizes RNA. It makes RNA. Okay, so let's think about this for a second. And we're thinking about initiation. How does this all start? So these questions here, or this, we'll try to fill in these answers as we go, okay? I'm not sure where exactly we are in my notes, but I think I can find it. All right, so the first thing that happens, here's our DNA molecule that needs to, and here's the gene that needs to be transcribed. So here's our gene that needs to be transcribed, okay? Now, what happens is RNA polymerase is an enzyme that recognizes the gene, binds to it. I'm going to draw it as a funny shaped triangle. That's RNA polymerase. I'm just going to write RNA pol. RNA polymerase recognizes the gene and binds to it and unwinds the double-stranded DNA. That's the first thing it has to do, so that it can transcribe the gene, right? So it partially unwinds the double-stranded DNA, and one of the strands then is going to become one of the strands of DNA becomes the template strand, okay? The strand that's used to make our copy. the template DNA strand here is running from 3' to 5'. That's important too. The directions are very important in DNA replication and also transcription and translation. What RNA polymerase does then, let's see. First, our DNA partially unwinds due to the enzyme RNA polymerase. Let's stick it in there. Okay. One of the strands becomes the template strand for RNA synthesis. RNA polymerase catalyzes the attachment of RNA nucleotides together. Okay, so what happens here is as RNA polymerase moves along this gene, it builds RNA. That's what it does. It builds the RNA. In other words, if it sees a T, on the DNA, it puts in an A. I'm doing something crazy here. If it sees a G, it puts in a C. Okay? It starts to build the RNA. Okay? And it builds it in one particular direction. It builds the RNA in a 5' to 3' direction on the RNA strand. So my 5' to 3' direction, my RNA molecule is going to grow this way. This is my 3' end over here. It's anti-parallel to the template DNA strand. Do you see that? Okay, we're going to keep thinking about this in our next couple of slides. RNA grows in the 5' to 3' direction. Where does transcription actually start? How does the RNA polymerase know where to start? It better start at the beginning of the gene, right? But how does it know where the beginning of a gene is? We have to talk about a promoter, all right? So when we look at a gene, let's say that this is our gene to be transcribed here, okay? At the beginning of it, at the front end of this gene, is a short sequence of DNA, maybe 25 base pairs, about 60 base pairs long. And that short sequence of DNA, actually I should have drawn this in green or something, okay? That short sequence of DNA upstream of the gene to be transcribed is called the promoter. Okay? Promoters are recognized by the transcription machinery. Now, promoters all have, I'm actually going to change to green here, promoters actually have a highly conserved sequence within them somewhere, usually about 25 base pairs upstream of the gene that needs to be transcribed. And that sequence is called the TATA box. There is a nucleotide sequence that is TA, TA, A, okay? That's a part of this bigger promoter in almost every promoter we see. And there are promoters for every gene on our DNA, right? So what happens now is the transcription machinery sees the promoter region and recognizes it and binds to that region, right? So what RNA polymerase does, RNA polymerase is actually not here yet. The first thing that sees the TATA box and the promoter are transcription factors. These are proteins that kind of are going to initiate transcription. In our drawing in our slides on the next figure, they're little purple circles. So transcription factors recognize the promoter and they bind to the promoter region. Those are called TFs or transcription factors. Okay? Those are proteins that recognize the promoter, they bind to the promoter, and then they draw in RNA polymerases. And RNA polymerase is like, oh, aha, this is where I need to start transcribing my gene. So RNA polymerase recognizes the promoter, and the transcription factors kind of tell RNA polymerase, come here, come here. So let's call this RNA polymerase, okay? And then transcription can begin. And then RNA polymerase is going to transcribe the gene in the 5' to 3' direction. So we need promoters that identify where the gene starts and that can draw in the transcription machinery. Okay, here's just a picture of that in green. That's why I said I wanted to draw it in green. There's our gene up here, okay, that needs to be transcribed. There's a template strand that's going to be used as the template for the copying process, and the non-template strand. The promoter here, though, is what the transcription factors see. They bind to it, and then they draw in RNA polymerase, and they say, this is where transcription has to start. Okay, so elongation of the RNA molecule. What does RNA polymerase actually do? And by the way, I have a number here. There are different kinds of RNA polymerases, like 3 or 4 different types. Don't worry about it. I just call it RNA polymerase, or a type of RNA polymerase. What does RNA polymerase do? It unwinds, let's just write it down again. It unwinds the DNA, and it makes or builds RNA nucleotides that complement the DNA template strand. Let's look at a picture of that. Ooh, I don't think I have that on here. Oh, this is a nice drawing. I should have just started with this one. So what's happening is, and I'll do colors, bright color choices. Suppose we have on the DNA template strand, suppose we have, you know, G, C, C, T, A, something like that. What I say, GCCTA, RNA polymerase will recognize those nucleotides and build the RNA that complements them. So if it sees GCCTA, what would it build? Well, if it sees a G, what would it put in place? It would put in place a C. If it sees a C, it'll put in a G. If it sees another C, it'll put in another G. If it sees a T, it'll put in an A. If it sees an A, it'll put in a T. Oh, not a T. It'll put in a U. Oh, sometimes I touch something the wrong way on this thing. It's crazy. All right? It's going to build RNA that complements the template DNA strand. And it's going to continue building in the 5' to 3' direction as it moves down the length of the gene. Okay? Notice that the RNA is building 5' to 3'. We're adding nucleotides to the 3' end of our growing RNA molecule. Okay? And what's interesting is, let me see. Yeah, let's finish these here. What's interesting